Welcome back to the second sessions of Symmetries and Morphogenesis. I hope you did enjoy the breakout rooms if you're not by yourselves there. This session we will address a fundamental question in developmental biology. How does the body axis develop during morphogenesis? Meaning, how can an embryo develop from a spherically or radically symmetric object into an elongated body that has two distinct poles, a head and a tail? We have two excellent speakers that will offer very exciting physical insight into this question. First, we will hear from Professor Kinneret Keren from the Technion University in Israel. Kinneret's research lies at the interface of physics and biology. Kinneret studies the self-organization of actin machinery in a wide range of systems and on a wide range of scales, from microscopic reconstituted systems to cell motility and tissue morphogenesis. And today she will tell us about the role of nematic order and defects of actin fibers in axis formation in Hydra. Before we begin, I would like to remind you that if you're interested in asking questions during the talk, please click the raise hand button and we will invite you to ask the question when the time is right. So without further ado, Kinneret, please take it away.
Thanks for organizing this nice webinar meeting. So I want to talk to you about our work on Hydra morphogenesis and how the polar axis is determined during regeneration. Morphogenesis arises from a complex interplay between signal pathways that involve patterning functional tissues and physical processes that ultimately shape the 3D form of the developing organism. And this involves multiple feedback on all levels of organization, from the molecular level all the way to the whole tissue. From a physical point of view, we have this far from equilibrium system, which is a many-body system with strong interactions, yet somehow this system is able to self-organize across scales to form very well-defined patterns. And one of the central questions in the field, as alluded to by Takashi, is how these systems are able to organize in such a robust manner. And an attractive approach to address this question is to seek a coarse-grained effective description of morphogenesis. This implies identifying relevant coarse-grained variables and developing a framework for understanding their dynamics. So in my talk today, I'll first introduce Hydra, which is the model system used to study this question. Hydra is a classic model system for morphogenesis. Many of the early developments in the field of morphogenesis were based on research on Hydra. And as I'll show you today, it's still a very valuable system to look at this interplay between mechanics and signaling pathways. In the first part of my talk, I'll talk about the supercellular organization of actin fibers in regenerating Hydra, and I'll show you the topological defects in the order of these nematic fibers actually serve as organization centers for the process of regeneration. In the second part of my talk, I'll focus on axis polarity, and I'll show you experiments where we generate frustrating initial conditions with respect to polarity and how these influence the regeneration and what we can learn about the development of the polar body axis in Hydra. In the last part, I would want to wrap up everything together and put forward the idea that the nematic actin fiber orientation field can act as a mechanical morphogen, and that the patterning of the regeneration process involves an interplay between this mechanical field and the signaling processes. The model organism we use for these studies is Hydra, which is a small multicellular freshwater animal. It's a member of the Nigerian family, which includes jellyfish and corals. All these have stinging cells, which they use to capture prey. The body plan on Hydra is very simple. It's basically a tubular structure with a head on one side and a foot on the other side. The head contains a mouth and tentacles that it uses to catch prey, and the foot allows it to attach to surfaces. The animal is about 1 centimeter in size. It contains about 100,000 cells. When you look in more detail into the structure, you see that this tubular body is actually composed of a bilayer of cells. This bilayer is composed of an outer layer, the ectoderm, and an inner layer, the endoderm. And in between, there's an extracellular matrix. These epithelial layers double as primitive muscle, and they're actomyosin, contractile actomyosin cables running all along the body axis in the ectoderm and in the circumferential direction in the endoderm. And I'll show you these fibers in a little while. What makes Hydra such a wonderful model system for looking at morphogenesis are its remarkable regeneration properties. So you can excise a small piece of tissue, only a few % of the whole body, or dissociate cells completely, and these will form a hollow spheroid, which will regenerate into a full animal within a couple of days. You can see this process in this movie here. So what you're seeing is a hollow spheroid that's formed from an excised tissue piece that regenerates over the course of less than 2 days to form this small animal. What makes this a really nice system is that we have a lot of flexibility. We can play with the initial condition of the system. The tissue is accessible for physical manipulation. It's relatively transparent, and we can follow the whole process, the whole regeneration process from start to finish. The main cytoskeletal feature in Hydra are these supracellular actin fibers. Here you can see them in a transgenic Hydra expressing LIFAC GFP. You can see that these fibers span the entire body from the foot of these animals all the way to the head. These are supracellular fibers. You can see they span multiple cells, and they're aligned in a very parallel array. This type of organization, where elongated objects align along the preferred orientation, is what's called a nematic organization, and you can characterize this alignment locally by a director field. So this is a headless vector that locally aligns with the average orientation of the fibers. Now, while the actin filaments themselves are polar filaments, these contractile actin fibers are composed of actin filaments in both orientations to generate contractions. So the fibers themselves are not polar, and they're described by this headless vector. You can see here the director field characterizing the orientation of the actin fibers in this region. We can extract these director fields computationally by looking at the image intensity gradients. So when we have such a parallel array, the gradients are largest perpendicular to the direction of these fibers, and we can use automated analysis similar to what people use in fingerprint analysis to extract these director field. This director field varies slowly in space and time. You can see an example of this here. What you're seeing here are snapshots from a movie taken a minute apart of a process of cell division. So there's this epithelial cell here that is dividing. You can see that these fibers remain stable across this process, and that they're maintain the structures that are much larger than individual cells. An important feature of nematic systems is the presence of defects. So defects are singularities in the director field. These point singularities can be characterized into different types of defects that are characterized by different charges. So you can see here +½ defects, -½ defects, and +1 defects. These charges are basically the winding number. As you go around the defect, this will tell you how many times the director field rotates. Another important aspect of these defects is that when you look at a closed shape, like the spheroids, Hydra is a spheroid, then the total charge is constrained by topology. So there will be a total charge of +2 on a spheroid, and you're all familiar with this. If you look at the globe, you know that if you look at longitudinal lines of the globe, you know that there has to be a north pole and a south pole. Each of these has a charge of +1 for a sum of +2. If we look at regenerating Hydra, these are the type of point defects we see in tissue. So we see +½ defects, -½ defects, and +1 defects. On the top here, you see projected images of the actin fibers in regenerating tissue spheroids. And on the bottom, you see the director fields, these black lines, and the color represents the nematic order parameter. This will be 1 when the fibers are well aligned, and we'll have a minima in regions where there's misalignment, which allows us to visualize very nicely the defects in the system. Now, importantly, when we look at the mature Hydra, we see that the topological defects coincide with the morphological features in the animal. So the defects occur in the tip of the head and the mouth, at the tip of the foot. We have +1 defects at each of these, and then each tentacle has a +1 defect at the tip and 2 -½ defects at the base of the tentacle. So altogether, this +1 defect at the head and the foot account for the topologically required total charge of +2, whereas each tentacle doesn't contribute because it has a net charge of 0. If we have cases of multi-axis Hydra, like you can see here in this 2-headed Hydra, then we have an extra head which carries an extra +1 defect. This will be balanced by 2 -½ defects at the junction. Now let's consider the regeneration process. So if we look at excised tissue pieces, they are excised from the body of the Hydra, and you see that the body has this beautiful parallel array of actin fibers. When we cut a piece of tissue, in this case a square fragment, it will inherit this organization of the fiber, and we have this parallel array in the excised tissue, which provides a structural memory of the original body axis. Now, during regeneration, the first thing that this flat piece of tissue does is fold into a spheroid. When it folds into the spheroid, the nematic orientation cannot be maintained everywhere. And indeed, when we look at these folded spheroids, here you can see an image of a spheroid done by light sheet where you see the front and back part of the same spheroid. You see that in half of the spheroid, you see this very nice array of actin fibers. This is the central part of the fragment where the fibers are maintained. Whereas in the other side, in the region of the closure, we see that the fibers have disappeared. Over time, we see a process where there's an induction of order. So this ordered region becomes ordered over time. You can see here the fraction of ordered area in spheroids as the function of time. And you see it increases during the first 24 hours due to this process of induction of order. So fibers appear in the disordered regions and they're induced by the inherited fibers in the ordered region. And this stabilizes at about 24 hours to a level close to 1. So now if you consider the full regeneration process, we start with this piece of tissue, a flat fragment that has the inherited fibers. It falls into a spheroid. Once we have the induction of order, point defects in this will emerge. There have to be defects because of the change in topology from a flat surface to a spheroid. And then over time, these defects will evolve, and they will end up in the characteristic defect configuration I showed you of the full animal, where the defects coincide with a morphological feature. This suggests that by following the defects from this folded spheroid to the regenerated animal, we can get an effective description of morphogenesis and hopefully a predictive description of this process. So let's look at how these dynamics look. So here where you can see...
Kinneret, I see there are some questions. Can we stop for a few questions or
okay,
later?
We can stop, sure.
Boris? So sorry to barge in with questions here, but so you just told us that when the spheroid closes up, order disappears.
Partially the order is maintained. So if I go back, you can also see it. So you can see here, you see this region is very well ordered. This region is ordered.
So how does it disappear? Where does it disappear? Does it disappear before the spheroid closes?
So
or
actually,
after it closes and it melts?
So actually what we see is that this appearance is correlated with the stretching of the tissues. When you have this fragment, in order to closes in a kind of purse string manner, the regions near the boundary of the fragment actually stretch a lot. And in these regions, we see highly disordered fibers. Whereas in the central part of the fragment, these are maintained. If, for example, we take two of these fragments and we put them together and use them so there's much less stretching going on, the fibers will remain in most of the regions, and only in the region of the adherence they will reorganize.
And stretching is really a deformation as opposed to a region of cell proliferation.
Yes, so all this process of morphogenesis can actually happen entirely without proliferation. So you can stop, classic experiments showed you can totally stop proliferation and you still get regeneration. It's not to say that there isn't cell division during normal regeneration, but on the time scale of this closure, it's all stretching of the cell. And indeed, you can see that the closure region becomes thinner.
Thank you very much. Thank you.
Can I go on, or are there further questions?
I don't see any question at the moment. So I think we can.
Okay. Okay, so in this movie, we can see the process of how the actin fibers organize during regeneration. What you're seeing here is a light sheet movie, and you're seeing it from 4 different directions. On the top, you're seeing the projected images from these 4 different orientations. On the bottom, you see the orientation field of the actin fibers, and the color indicates the nematic order parameter. You saw early on that there were disordered regions. These have disappeared. By now, the system has order everywhere, and we see that point defects emerge. You see that these defects are stable over time, but there's a lot of dynamics. They move relative to the tissue, and the configuration of defects evolved until by now we can see the typical configuration of a mature Hydra. You can see the head here with the tentacles forming and the foot here with the +1 defects at the head and the foot. So if you look at the final configuration, we have the +1 defect at the foot and the +1 defect at the mouth in this process. So we can characterize the evolution of the nematic field and the defect configuration in the tissue over time. And you can see this here. So what you're seeing here is the defects of the different types as a function of time averaged over different samples. And what you can see is that initially there's a large disordered region, so there are no point defects. Over time, as the system orders, we have point defects emerge. The most common defect early on are the +half defects. These are actually entirely absent from the mature animal, and indeed we see that over time the +half defects disappear, and we see that the +1 defects become get to the value of 2, which characterize the +1 at the head and the foot. If we look at the net charge, during this induction of order, as the disordered regions become ordered, we see that the charge of the point defect increases to the topologically required level of 2, and then it remains stable at 2. During this time, you can see the defect configuration evolves, but the total charge is constrained to 2. If we look more closely at the dynamics of the system, we can see that the +half defects are actually mobile. To visualize this, we uncage a fluorescent dye to label cells in a particular region. And as you can see at these images from a movie, you can see that the +half defect is moving towards its round end away from these labeled cells. Over time, the +half defects disappear. This occurs primarily by merging of 2 defects, as you can see here. So these 2 +half defects facing each other merge to become a +1 defect. Or we also sometimes see annihilation of defects where a +half defect moves into a -half defect and annihilates. What's striking is that the defect configuration and its dynamics are closely related to the morphogenesis process and the formation of morphological features. So when we see these 2 half defects merge to form a +1 defect, this will predominantly become the foot of the regenerated animal. So the 2 +half defects meet, and this meeting point is the site where the foot will form. When we find +1 defects early on in the regeneration, these defects actually remain extremely stable relative to the underlying cells. So you see here again labeled cells, and you can see that the +1 defect is not moving relative to the labeled cells, and these sites will predominantly become the head of the animal. So this +1 defect becomes the tip of the mouth in the regenerated animal. I want to emphasize that by looking at only at the dynamics of the actin fiber orientation field, we can identify the sites that will become the foot and the head very early on, way before any structures or cell differentiations occur in these regions. So how are these topological defects related to the formation of the functional features? Let's focus on the head. So I showed you that when we observed early +1 defects, in this case, this is 4 hours after excision, these will remain stable relative to the underlying cells, and the cells in this region will eventually form the mouth, the tip of the head of the regenerated animal. Now, to form a mouth, there has to be significant changes in the biology of the system, in the morphology and differentiation. What's special about these +1 defects that the cells in this site are the ones that do this process. So one obvious feature that is special about these defects, it comes directly from their structure as a focal point of contractile fibers, is the mechanical environment there. So these esters of contractile fibers, these fibers are involved in cell-cell adhesion, these through cell junctions. At the core of the defect, there are no fibers spanning the core, so we expect there to be weaker adhesion. And when all these contractile fibers contract, this region at the core of the defect will be under high tension. This is nicely reflected in the formation of rupture holes. The Hydra tissue has this gastric cavity inside, and it undergoes continuous inflation and rupture during regeneration, as you can see here in this trace of area versus time. And these ruptures correspond to hole formation in the tissue. And when we look where these holes are forming, we see that they form predominantly at the +1 defect site, as you can see in this image sequence. So there's never any holes that are formed at the ordered regions. All the holes appear at these +1 defect sites, indicating the special mechanical environment at this region. So what I've shown you is that the +1 defects appear early in the regeneration, and they remain stable with respect to the underlying cells. The cells in this region experience a unique mechanical environment, which comes from their structure of these defects as a core of aster of contractile fibers. This is reflected in the formation of holes at these +1 defect sites, and eventually this region becomes the head of the regenerated animal. So what this implies is that the region of the +1 defects act as organization centers for head formation. So these sites are the sites where all the action is happening, and eventually the head will form. So to summarize the first part of my talk, I've shown you that the actin orientation field provides a coarse-grained field, and we can follow the dynamics of this field, and active nematics provides a useful predictive framework for characterizing the dynamics of this orientation field in regenerating Hydra. And moreover, I showed you that +1 defect that are identified early in the tissue remain stable, and these sites are the sites where the morphological features appear. So this, I think, is a good time to stop for questions before I move to the second part.
David?
This is very, very beautiful, but presumably there's something going on with the cells themselves. This is an extracellular fiber thing, but the cells, they have a viscoelastic properties. Are there disclinations in the cells as well? Topologically, there have to be. And are they correlated with the defects that you are seeing in these fibers?
Mm-hmm. So actually, if you looked at the movie, you saw that there's a lot of dynamics in the tissue.
Yes.
So that actually, if you look at cell shape, that's a very variable quantity. So the shapes of the cells or the nematic orientation of the cell shape is not something stable. The fibers are very stable over the course of hours, but the cell shape as the tissue contracts and changes actually change with seconds and minutes differences. So it's actually very hard to look at the nematic, or let me put it, if I looked at the nematic orientation of the cell shapes, that would be a highly fluctuating field.
There's also an issue of coordination number of the cells. That's highly disordered. Lots of 7s and 5s and 6s and stuff like that.
We are looking into that now. It's relatively complicated because of the dynamics of the tissue and our ability to visualize that. I hope to have a question for that in the next few months. So,
but then the last question is it a viscous, can we think of it as a viscoelastic cellular medium that the cells flow freely? Or do you know what the viscosity is? Can you estimate it?
So actually, when we look at the movement of cells, you'll see it partly in my second part of the talk. There are cell movements and there are cell rearrangements, but there aren't dramatic flows during this elongation or these dynamics. So the tissue is certainly viscoelastic. There's not much cell movement. There is basically enough cell movement to accommodate the changes we see in the overall shape of the tissue, but there aren't dramatic flows of cells within.
Okay, then finally, when a tentacle is going to grow out.
Sorry to interrupt, but maybe we should take one more question from Christina and then leave the rest of the questions to them. Thank you.
I think David Lubensky was before me, though.
Oh, sorry.
Sorry.
So go ahead, Christina, that's okay.
Fairness. Oh, I just had a question. So it seems like during the process where initially you said you have +1F, and then at least two of them form this stable configuration that looks like a +1 from a distance. And during this process, your spheroid remains a spheroid. It doesn't get elongated yet. Is that true?
Actually, no. Actually, the elongation process and the movement of the 2 + ½ defects and their merger actually occur simultaneously. So we see the 2 + ½ defects moving towards the region that will become the foot as the tissue elongates.
Ah, okay. The reason I was wondering is because, as you know, in microtubule suspensions confined to a vesicle, the 4 + 1F seem to oscillate rather than coming together in stable configuration. So I was wondering what's the difference from, I don't expect you to know, but it does elongate, you're saying. Okay, thank you.
So maybe we'll continue the talk and leave the questions to the end.
So I'll keep going. So in the second part of my talk, I want to talk about how body axis polarity reorganizes in these regenerating tissues. In the first part of my talk, I showed you that the alignment of the body axis, there's a structural memory in the orientation of the actin fibers, which helps define the alignment of the body axis. But these contractile fibers, they're not polar, so it's not clear what defines the polarity of the regenerated animal. And I want to remind you that in these fragments, as they fold into a spheroid, this polarity cannot be inherited trivially. We did see that there is asymmetry in the configuration of the defects, but where the +1 defects are associated with head formation and the 2 +½ defects are associated with foot formation, but it's again unclear what gives rise to this polarity. It's also important to note that in Hydra, it's known for a very long time that there is a strong memory of polarity during regeneration. This is already evident in the classic experiments done by Tremblay in the 18th century, where he cut an animal in half and he saw that the head grows a foot and the foot grows a head. You can also cut a piece of tissue, a ring of tissue. So the ring will have a head-facing side and a foot-facing side, and invariably the head-facing side will grow the head and the foot will grow a foot. You can see an example of that in our experiments here, where we put a ring on a wire where we could label the foot side of the wire, and invariably we got the ring maintains its polarity. Now I want to emphasize that the ability to form a head is present in all the tissues. So wherever you cut the ring, the tissue can form in the gastric region, in the central part of the animal. The tissue can form a head. Yet when you take this ring, a head forms at the originally head-facing side of the ring. Now we're interested in how polarity is determined during regeneration. So it's useful to be able to perturb that in a controlled manner. And for that, we developed a system of ring doublets, where what we do is we take rings from two animals. So we take the rings from two separate animals, and now we can vary the relative polarity and the position of these two rings and fuse them into a ring doublet. So here you can see an example of the head-to-head configuration. So we take two rings and we adhere them on their head-facing side. So we obtain a doublet. At the edges of this doublet, it closes like a single ring at these two places, whereas at the middle, it adheres along the ring to the other ring, forming these doublets that you see here. And we can also vary the position of where we take the ring along the body axis. And using this configuration, we can now ask, how does the system reorganize under frustrating conditions? And this allows us to explore the process of establishing the body axis polarity. So when we take these head-to-head ring doublets and we look at their regeneration, what we see is that oftentimes you have a head in the middle. This is what you'd expect based on the original polarity of the tissues. You can see here a movie of that. You can see the two rings, they're forming this doublet. You can see the interface remains very well defined. There's no dramatic flows of cells in the tissue. And you can see that the head starts forming in the middle, generating this initially the bump. And you see the head form exactly at the middle between the two doublets, and you see two functional feet at both sides. But we can see that actually the polarity can also be reversed. So we also have different outcomes. For example, we see this polarity reversal. So in this case, we start off in the same configuration, but what we see is that initially the tissue looks very similar. We see this cylindrical animal, but in this case, we have a head forming, and it's forming actually on the foot-facing side at the edge of the tissue. So this is the place that, from a polarity point of view, is surprising because it's the furthest away from the head in the original animal, yet this is the site where the head forms. We can look at the dependence of polarity reorganization and also on the where the ring comes from the tissue. To do that, we use the head-to-foot configuration. So in this configuration, we take the two rings and we fuse them together while maintaining their orientation. But now what we do is we vary the original position of the ring. So we can take rings from above the midline, these are these upper rings, or rings from below the midline, these are these lower rings. And then we can combine them in an oriented manner where the lower ring is below the upper ring. So this is the same orientation as they were in the original animal. Or we can combine them in an opposite orientation, so in an anti-oriented configuration, where the upper ring is below the lower ring. So in both cases, the tissue has a preferred polarity. it has an easy axis for polarity along the original axis, but there are differences in where the tissue comes from the original body axis. When we look at the oriented configuration, we see that invariably we grow normal animals. So a head formation is suppressed in the middle, and the free head side will grow ahead, while maintaining the polarity of both rings. When we look at the anti-oriented configuration, we see that the regeneration outcomes are very variable. So the system has much more trouble to establish polarity. In some cases, we have a polarity on head growing on the original head-facing sides, but in other cases, we have heads in the middle, and we even have situations where both rings flip polarity. So let's focus on this, this configuration. So we have our rings, and what we see is that in this case, the system chooses to grow its head from the foot-facing side and flip polarity in both of the rings. So rather than grow along its easy axis and grow ahead from the originally head-facing sides, we see it flips both rings, and we have a head growing from originally foot-facing sides. So these experiments indicate that the polarity is labile, it can be changed, and it also demonstrates the importance of the original location along the body axis of the animal. So we have these three properties: the original polarity of the ring, the original position along the body axis, and the structure. So if we look at a simple configuration, like a single regenerating ring, all these are commensurate. So we have the polarity, the most head-facing side of the tissue, and the structure at the edge. All of these lead to the formation of the head at the end. However, in our ring doublets, we see that we can make these different properties non-commensurate, and we see that different things happen in these situations. So we see that head formation can occur on an originally foot-facing side, which would never occur in a single ring. And we see very commonly head suppression in the middle. So the structure in the middle is less letting the head grow, and it's often suppressing the head formation at the middle. So this emphasizes how all these different important factors play a role in establishing polarity in the regenerated animal. Now let's focus on this difference in the structure between the middle and the edge of the tissue. Going back to the actin fiber organization, there's a very clear difference between the edge and the middle with respect to the orientation of the actin fibers and the defects that we have. So when we look at an edge will seal and form a region with a net charge of +1, as you see here. Whereas at the middle, as the two rings seal to each other, we'll have a region with 0 net charge. So you can see an example of the doublet. The upper part has an edge with a +1 defect, and you see the nice connection between the two regions in the middle. So this gives us the actin fiber organization clearly differentiates between the middle and the edge. And now relating to what I showed you in the early part of my talk, when we look at these cases of the polarity reversal, so we are in the head-to-head configuration, and we are in the cases where a head grows on a originally foot-facing side of the tissue. You can see here images from a time-lapse movie of such a ring doublet. On the top, you see frames where the tissue, you can have a side view of the tissue, whereas in the bottom, I've shown you images from the same movie where we see a top view of the images. You see that this +1 defect forms very early on as the tissue seals at the edge. And you see that this exactly will be the site where the mouth will form when the head forms on the foot-facing side. So we have suppression of head formation at the middle, even though signaling-wise, we'd expect the head to form in the middle. The head does not form in the middle and doesn't form randomly in the tissue. It forms exactly at the +1 defect at the edge of the tissue. Now, as I showed you before, this is not always the case. In some cases, we have this polarity reversal. So the signaling for polarity in the middle is somehow suppressed and a new head forms at the +1 defect. In other cases, as you can see here, we have a head forming in the middle, and you can see here that the interface is smooth, and we have de novo defect formation, like you see in budding Hydra, at the middle to form the head in the middle. So this is a very nice demonstration of the interplay between signaling and mechanics that comes this process of polarity determination in these regenerating ring doublets.
Kinneret, we should probably conclude soon. Oh, okay, sorry. Jumped ahead.
Yes, I'm concluding. So to summarize this second part, we see that these ring doublets provide a very nice system to look at reorganization of polarity because we can engineer frustrated initial conditions. We see that the body axis polarity under these constraints is actually level and can even reverse. And we see that the emergence of polarity depends on the initial polarity of the tissue, the position along the axis, as well as the structure. And we can see the +1 defects act as attractors for head formations. So when we have a case of polarity reversal, the +1 defect will be the site of the new mouth. And I want to emphasize, this does not imply that the +1 defect is neither required nor sufficient for head formation. I showed you that the head can form from the middle, and in this case involves de novo defect generation. And also when we look at the foot side of the ring, it doesn't form a head even though you have a +1 defect. Nevertheless, it looks like the +1 defect acts as an attractor because the head forms, when it doesn't form where you expect based on polarity, it forms at the +1 defect. So to wrap the two parts of my talk together, I've shown you that the nematic orientation of the supracellular fibers defines a coarse-grained field. And I showed you that we can look at the dynamics of this coarse-grained field and obtain a predictive effective description of the regeneration process. And I've shown you that the process of regeneration, actually, this is not the only, obviously not the only thing that's happening, but there's actually an interplay between the biochemical signaling and mechanics. And this is nicely reflected in looking at the regeneration in these ring doublets, where we see this competition between signaling and this interplay between signaling and mechanics. And finally, I'd like to put forward this idea of thinking of the nematic actin orientation field as a mechanical morphogen, a mechanical analog of the chemical morphogens we're all familiar with. So we have here a field. This field varies in space and time, and it provides local cues to the system, to the cells in the system. So the cells will respond to the unique environment around defect sites. So the notion I want to put forward is that this mechanical morphogen interacting with other mechanical factors and biochemical morphogens in the system together lead to the robust formation of functional tissues in the regenerating hydra. So finally, I'd just like to thank my coworkers on this project. So this has been a long-standing close collaboration with the lab of Professor Erez Braun, who's my colleague at the physics department at the Technion. The first part of the talk was headed by Yonitze Moulezak, who's a graduate student in my lab. And the second part was a joint effort by Anton Lifshitz and the other Darion firm who are in my lab. And I'll also advertise I have open postdoc positions. If anyone is interested, please contact me. And thanks, and I'm happy to take questions.
Thank you, Kinneret, for this great talk. We have a bunch of questions. Dave Lubensky first.
Hi, that was a really fascinating talk. So I was wondering if you could say a little bit more about what's known about the cell biology of these actin fibers. Are they anchored in some particular way at cell-cell junctions so that so that they stay connected across the junctions? When they move, are they moving from one cell to another, or is the cell moving with the fiber? And also in particular, in the defect cores, how many cells is that, and are the cells arranged in a particular way?
So these fibers, these are contractile actomyosin fibers, similar to what you'd have perhaps in a smooth muscle. They're connected between cells through cell-cell junction and also adhere to the extracellular matrix, to the mesoglia. So they appear, what you saw in all my images, they appear to be continuous across a very long scale, but it's actually fibers from individual cells. In the case of the ectoderm, they actually protrude about a cell length in each direction. So there's an overlap of fibers between neighboring cells. We see that as the tissue contracts and changes shape, these fibers remain stable, but they're certainly able to reorganize. For example, that's most obvious when there's a disordered region near the closure region, or when you take cell aggregates, and these fibers will organize spontaneously to form these parallel arrays. And what was your second question?
So to follow up on what you just said, so that means there's not actually one really continuous fiber that goes the whole length of the hydra.
Mechanically, it's a continuous fiber, but it's built from overlapping fibers from individual cells.
Right, so the second part was particularly about the structure of the defect cores. Just what's the scale in terms of cells, and if there's rupture in them, does that imply the cells are arranged in a particular way?
So, so the defects, if you look at the mature animals, the minus half defect, the plus half defect, you saw in the images, they're quite well localized, and the typical dimensions of this is similar to cellular dimensions of the core of the defect. The plus one defect, it actually depends. So the plus one defect at the mouth is actually very beautiful line. And there actually there's the mouth is functionally opens and closes, and the cells are organized. So the core of the defect is kind of in between the cells. So when it opens, it doesn't rupture cells, but it's in between the cells. At the foot, the plus one defect, it's an effective plus one defect. There's actually a basal disc that the animal uses to attach to surfaces, so it's not exactly a close aster.
Okay, excellent. Thank you.
Yeah.
Yes, hi. I had a very simple question, rather a confusion really, which is if I take a nematic and then I do know that the ordered phase is the lowest energy phase, of course, but then the low-lying excitations are where the directed field is continuous. And the classic understanding of defects is that there are hard constraints which actually impose a geometric constraint which actually cause these defects to form. And I'm rather curious that this is really not a hard constraint, it's a soft material. And yet in these systems, you happen to generate these kind of defects. Is there an understanding why this might happen? Why, for example, the defect isn't, it's not possible to rearrange these and get rid of the defects?
I mean, getting rid of the defects in this system is not possible because of the topology. So you're constrained to have a +2 total charge. How this +2 total charge is distributed, if you have 4 plus half defects, or you have 2 plus 1 defects, or you have negative defects, that's another question. But you're constrained to have some defects if the whole system organizes nematically in a closed system.
Yeah, but you don't start off with having a +2 charge. Is that?
I start off, so what I start off, like for example, in these fragments, I start off with a region that is totally ordered. There are no defects, and then there's a large disordered region there's a large disordered region around the closure region. Now, as the system, so the first thing that the system does is it induces order. So you have nematic order that emerges in all the system. Once you have this nematic order in all the system, now you're constrained to have the defects. I agree with you. Have this ordered region, they're not point defects, but once the system does organize, they become point defects, and then you're constrained to have a total charge of 2.
Okay, maybe we can discuss this later on. Okay, thank you.
Boris, I think last question, and then we'll have to move to the next talk.
So thank you for a wonderful talk. Could you elaborate a little bit on that coupling between nematic order and biochemical signaling? Presumably WNT is a conventional morphogen that, remember reading in Walford's book?
Yeah. So I mean, I think that when you look at the classic notion in Hydra is that there's the main feature that determines polarity is the head organizer, and this head organizer is characterized by having a WNT at the tip of the mouse, and it's autocatalytic. And when you take, a lot of the studies are done on bisected animal. When you take a bisected animal, then all these the structure and the chemical factors are all commensurate, and you have the WNT signaling emerging at the tip. However, in more complicated more complicated things, the development is not so trivial, and these factors can be non-commensurate, like we saw in the ring doublet experiments. Now, what we know is that there's, at the end of the day, the animal will have a very clear polarity, and the chemical morphogen, the WNTs, will be organized along this axis, and the actin fibers will be well organized along this axis. What our experiments suggest is that during the regeneration, that the establishment of the body axis in polarity is a dynamic process, and these signaling molecules, they arise when you make a wound. There's a wound response, and many signals arise. But what we're suggesting is that the presence of defects in the right context can influence, can help in this self-organization that leads to the formation of the body axis. Now, everything I'm saying is not mechanistic. We don't have any, I mean, it's known that winds can influence actin organization for sure, and it's known that actin organization can influence winds via mechanotransduction. So there's a mechanistic insight that these two are talking to each other, but what's actually going on here, we don't have any, this is, room for further research.
So you didn't, for example, observe these +1 defect ruptures as sources of wind?
So we don't have the ability to look at wind live. The only markers we have for wind are GFP with a wind promoter, and that gives a signal way after you see the hypostone form and all the morphological features. So we cannot see what is happening early. But this is certainly something we're interested to know. And what I would guess is that these defects actually are sites where wind will be enhanced in some way compared to other regions and lead to the formation of an organizer.
Thank you.
One last question by Matan. Matan? Okay, he disappeared. Matthias?
Can you hear me?
Yes. Thank you.
Okay.
Thanks, Kinneret, for the talk. So, Matan, do you want to?
Can you hear me?
Maybe. Yes, go ahead.
Excellent. I have a question about the energetics of this. So in a nematic texture, each defect has an associated elastic energy. And on a closed surface, you get 2 + 1 defects just by topology. But in principle, you can get more of them, especially in a finite temperature. So I don't know what is the energetics and what is the elastic energy of these fibers, but can you estimate it? And maybe from this, can you predict rates of budding or rates of forming extra feet?
So first of all, I can't, but what I can say is that you can see in our system, so apart from forming tentacles, which is a process that happens later, if you look at the early stages where you establish the polar body axis, we actually see that our system has the kind of very small number of defects, so it's a very small system. And we see that these defects, we don't see any emergence of new defects in the system. So we see the defects forming only in the original disordered regions. And once these defects are formed, then they have some evolution, but we don't see new defects forming. So in that sense, it would be what you say a low temperature. We don't see, for example, many defects emerging at these stages with respect to the body axis forming state.
Okay, thank you again, Kinneret, for the great talk, and thank you everybody for the questions. I see there are more questions, but maybe you can continue that offline. We will